E ka mana e ngā reo o rangatirama tēnā koutou katoa. E mihi ana ki te mana whenua me ngā tangata whenua o Tahiti, tēnā koutou. Tātou katoa e huihui mai nei tēnā koutou, tēnā tātou katoa. It's lovely to be here. And we've made it to the last talk of the day. It's been particularly nice to get together with people after such a long time when we couldn't get together face to face. So I thank the organizers of this meeting and look forward to the discussions that I hope we will continue to have over the next day or so. It's also particularly nice for me to be here in Tahiti, as it's almost exactly 29 years to the day that I first came here to do my field work, my first outside of Aotearoa field work, trapping rats throughout the island, throughout Tahiti, Raiatea, Huahine, and Wahuka, where I met Eric. And we talked about this crazy rat woman who was looking at rat DNA. So that was November in 1993. And it was in 1994 that I published the first paper kind of describing the development of the commensal model and looking at mitochondrial DNA variation in Rattus excellens as a proxy for tracking the movement of the waka, initially through Polynesia, but as the years have progressed, we've expanded that approach. So the Kiore, or Rattus excellens, Iore, is the most widely dispersed of the commensal animals in the Pacific. It is found in the earliest archaeological deposits, and most importantly, I think, as Catherine spoke on the first day, the fact that it does not interbreed with the European introduced rats. So the rats that we find on the islands today are the descendants of those that were introduced by the first colonists, and we've now been able to see this in both the modern data in comparison with the early archaeological remains of Kiore. So that first study, which was really my PhD research, looked specifically at extant populations of Rattus excellens from throughout Polynesia, and we identified two interaction spheres. Keep an eye on the time. With a central Hawaii zone that incorporated the Society Islands and the Southern Cook Islands. So we had the southern interaction zone where we saw evidence of multiple introductions of rats from this zone to Aotearoa, with the Kermadec Islands acting as a stepping stone, with what appeared to be a kind of single introduction to the Chatham Islands. And then we had a northern interaction sphere that incorporated the Marquesas, Hawaii, and this Hawaii zone and the societies and cooks. We then, with the development of methodologies, found that we could extend the model further into the Pacific using both extant, living, recently killed rats, but also ancient DNA and DNA from museum collections. And so we extended the sampling across the Pacific into Near Oceania to try to understand and address the question about the origins of the Lepida rats, for example, and up into island Southeast Asia. And we found three very distinct haplogroups, very clearly defined geographically. Group one that we have here in the Philippines through Borneo and Sulawesi. Group two, which again incorporates the Philippines through the Reef Santa Cruz, and then the remote oceanic rats, which we saw two kind of subgroups, which we referred to as 3A and 3B. But again, this was using a small fragment of the mitochondrial DNA that gave us these patterns. There was not much variation within remote Oceania. We did have these kind of two groups within group three, but the majority of our samples belong to this central haplotype we referred to as R9. We were also able to expand the commensal model beyond the rats to look at the other animals that were transported in the waka that came out into the Pacific. So we looked at pigs, chickens, and started looking at dogs to see whether we saw similar patterns. And of course, for these commensal animals, we had to work with archaeological remains because the populations of pigs and dogs and chickens that we see on the islands today are admixed with the European breeds that have been introduced. So working with a number of colleagues overseas, we found, looking at ancient pig DNA, that there was a single clade, a single group of pigs that were found throughout the Pacific. And we could trace the origin of that Pacific clade, not to Taiwan or the Philippines, but back to mainland Asia, the low frequency and of this haplotype in pigs from Vietnam. We also started looking at the chicken bone, and there was a significant amount of chicken bone from the Pacific. And this is the work of my PhD student, Alice Story. And while she was investigating mitochondrial DNA variation in chickens in the Pacific, we were approached with some possible chicken bone, possible pre-contact, pre-Columbian chicken bone from Chile. And we were not expecting to see Pacific chickens in South America from the site of El Arenal. But as Anna discussed yesterday, the sequences that we got from the first sample came back as identical to chicken bones that we had sequenced from Melehavea in Tonga and Patumaputi in American Samoa. And we then had that result replicated and had a radiocarbon date taken from that same bone, and it produced a clearly pre-Columbian date. We then sequenced 2 more bones from the site and radiocarbon dated them, and they came back with very similar DNA results. So we argued that this was evidence that Polynesians certainly made it all the way to the Americas and left the chicken but brought back the kumara. So a swap of chicken for chips, which sounds like it was a pretty good idea. And it was the earlier lineage we saw in looking at the Pacific chickens, 2 different lineages in Polynesia. What we considered an early one that was similar to the Tongan and Samoan material, and a later lineage that we see in Hawaii and Rapa Nui. And it was the earlier lineage that was taken to the Americas. But it was the dogs actually that were the most interesting in a way when we started looking at expanding to dog DNA and looking at early Pacific dog. And going through collections, we found there's very little dog, if any, clear Lepida-associated dog in the Pacific. So, but lots of dog remains in later sites. So after 1500 BP, there's lots of dog in Polynesia. But interestingly, and I'd love to know if this still holds, no prehistoric dog in New Caledonia or Vanuatu, which was confusing to us or surprising to us in a way, thinking, well, how did dogs get out here if they're not coming through Vanuatu and New Caledonia? And this is when David Addison and I, an archaeologist who's working on Samoa, American Samoa, proposed our crazy idea that maybe there was kind of going back to Te Rangihiroa, another movement of people that were not passing exclusively through Micronesia, but were visible in their movement through Micronesia because the islands had not, many of the islands had not yet been settled. And this was information that we were pretty excited to see via Bill Ayres, who was showing us these maps about the final sea level drop about 2000 BP. This was the picture, 5 minutes, at 3000 BP, and then at 2000 BP, all of the atolls are exposed. So we thought that there was perhaps a movement of people that were inspired by this, this kind of, in a sense, stepping stone of islands across the Pacific. And interestingly, when we proposed that in 2010, lots of people told us we were crazy and we said we don't know what's going on, but here's just a possible explanation of a kind of multiple migrations through the Pacific. And this is a paper from 2020 modeling using human genome data, whole genome sequencing of over almost 1,200 people. This is a paper that Ethan was involved with that shows a kind of slow population growth and then a rapid increase in population growth at about 1,000 years ago, followed by a population crash with the introduction of European diseases. So this fits with the possibility of another population migration out. We've also been working within New Zealand, sampling Maori and Pacific peoples in Aotearoa, and we started finding that there were clear differences between East Polynesian and West Polynesian human genomes. This is using an Ancestry 750K ancestry chip data. But East Polynesians, Maori and Cook Islands, clearly different from West Polynesians, Samoan, Tongans, and Niueans. And when we compared our data from both modern and ancient New Zealand samples to the data that had been generated by Cosimo and Skoglund and Hujishoff and so forth, you can see that here are the East Polynesians, here are the West Polynesians in red, and the Polynesian outliers in pink here. The Lepida crania from Teuma and Tonga are just there. So we see distinct variation there. When we continue to look at dog mitogenomes, complete mitogenomes from ancient dogs and modern dingoes and New Guinea singing dogs, this is work of Karen Gregg and colleagues, we find, again, complete mitogenome, a single haplogroup of dogs in remote Oceania. We see them coming in late. This is a Taurama puppy from New Guinea, and in the Torres Strait Islands. But interestingly, there are dogs coming down from Taiwan into New Guinea singing dogs and dingoes trace back to ancient dogs from Taiwan. And then we have another lineage, the B lineage of dogs from Taiwan that we're also finding along the south coast in New Guinea, and yet another dog lineage in Timor. So things are really complicated with dogs, but late dogs coming out into Polynesia and remote Oceania. So the patterns that we're seeing consistently, 2 introductions of rats coming out into remote Pacific, multiple introductions of dogs, but only one of those comes out into the remote Pacific, and that is sometime after 1500 BP. We still see 2 introductions of chickens. So there are key questions that are identified through this combination of commensal data. What's happening with pre-Lepida interactions in island Southeast Asia and near Oceania? What about the Lepida, those Lepida results from Teuma? Where are those canoes coming from? We've got late Lepida and post-Lepida arrivals in near and remote Oceania. What's going on at 1500 BP? What's causing the East-West Polynesian differences that we're seeing in humans? What's the relationship between the Polynesian outliers and East and West Polynesia? And some of the islands like the Phoenix and Lion Islands. We've actually got some rats. We were hoping to have that data this week, but we had a problem with the sequencing run. We're also interested in looking at variation within Polynesia and site or archipelago specific questions. So we're getting some of these interesting results with the chicken results that Anna spoke about, where we have these 2 groups of chickens with early Tongan chickens with Yap and Chook, and also coming into Hawaii, and then these other introductions into East Polynesia. And similarly with the rats, we got some very exciting results that are 2 completely different groups of rats within East Polynesia, the societies Aotearoa, Cook Islands, Mangareva, Marquesan, but also the Marquesan samples down here with Rapa Nui. And then we've got the Rekohu, or the Chatham Islands, Kiore, ancient and modern, off here on their own. So we're very interested to see where those Kiore are coming from. Very keen on looking at the Austral Islands and getting some more samples from French Polynesia. But some other interesting things, what's going on here with Rotuma. These are ancient Rotuman rats here with the modern Pukapukan and PNG. So with a central Western Polynesians and Fiji cluster there. So just an exciting future, I think, for commensal studies. We are continuing to analyze more commensal samples, ideally from well-dated contexts. We're getting now genome-wide data from ancient and modern Kiore, ancient chickens, and potentially pigs and dogs and other commensals. As we've heard in the previous paper, there are new commensals. There are new methods that we can try. We have a student who did some bulk bone metabar coding of some Sue Bulmer samples from Kiowa, and it pulled some pig DNA out of the layers that Sue Bulmer was confident had pigs. So we're following up on that. Interesting thinking about the commensals of the commensals, so things like coprolite analysis and dogs. And we're also working on a project with colleagues from Guam and the University of Papua New Guinea, looking at whole genome sequencing of people to try to reconstruct and understand the evolution of Pacific genomes and the implications of health for Pacific people. So putting all of this information together. Just like to acknowledge funding and institutional support, all of the archaeologists who have provided us with samples, many of you here already, the communities who have collaborated with us, and Maruroa for everything and the support we've had with our translators and everybody to make this conference possible. Thank you.